Hello, Sharon. Hello, Professor Sharon Schroeder, March 29th. Lab 4 microscopy for microbiology. This is exercise 2, where I'm going to be taking a look at observing microbes. It's gaining proficiency in observing microbes. So I have all my slides here laid out. So we're going to be looking at amoeba, an amoeba whole mount, penicillium with candida, yeast, whole mount, bacteria, spirillum, bacteria, bacillus form, and bacteria coccus in coccus form. So I'm going to go ahead and start with the amoeba. So let me get this. So immediately I see that I need to adjust where the little, it's like a little circular space. This is where the specimen is. So I have to adjust that. Let me see here. Okay, so first I'm going to view it under low power. That's where we usually want to start. Okay. So I'm going to go down. Alrighty. And as you had suggested, Professor, going slower does help. How far am I though? Okay. So immediately I see a little red splotches. Let me kind of fine tune this a little bit to see if I can. And my eyes are really horrible, so this is a little challenging for me. So I have already, I'm doing double work because I can't, like I said, take pictures and do a video. So I've already taken pictures of these slides. But I also, I mean, it's taken me a long time to do this assignment, but I feel like I'm getting better because I've used the microscope, I would say, a little bit longer. So yeah, I see a couple of red splotches. This is for the amoeba that I'm looking at. Let me make sure that I don't need to do anything else. So now it's asking to switch to medium. So that was pretty cool looking at it from just the 4X. So now I went ahead and switched it over to the medium power. So I'm gonna go ahead and take a look here. Oh yeah. Now the little microbes, they look bigger in size. Let me see if I can get any like detailed. It's a deep red. Okay. So that's pretty cool. They're pretty big. Replace, okay. Now, of course, switch the objective lens to the high power. So let me go ahead and do that on the microbe. Now for the high power, I'm not seeing that vibrant red color. that I saw with the 4X or the medium power. Let's see here. Yeah, I don't see the color. It looks clear, like you can clearly see that there's something smeared on the slide, but it doesn't really have a color. It just looks kind of grayish clear. So my observation is the medium powered lens so far is the one that I can see the specimen the best on. So now it's asking to add a so replace the lowest level with the oil immersion lens. I'm gonna go ahead and do that. And then for this exercise, oh shucks, good thing I caught that. This is gonna go down with, this is how it's gonna go with each of the different specimens here. So let me put this on. I'm going to add a drop of the immersion oil. I'm going to switch this over to the oil immersion lens. Okay. So let me move this over a little bit to make sure the oil has the light going through it. There we go. Very good. Okay, so let me go ahead and take a look. This one I know that I have to pretty much get the lens into the oil to view what I need. So again, I'm not sure if I'm supposed to be viewing different colors of specimen. I don't see any color on this. It's just gray, like a grayish background with some black spots. Again, you can clearly tell that there's something on the slide, but what it is, I can't differentiate. Yeah, that's what I'm getting for the oil immersion lens for this slide. So I'm going to go ahead and take this lens off and replace it back with the 10X. I'm sorry, the 4X. I'm going to clean this with my lens paper really quickly. Okay. Removing this slide. And then I'm going to go ahead and just go down the line, explaining each one of what I see. Forgive me. Getting these lenses in is a little challenging sometimes. So the next one, let me make sure. So I took a photo already. I uploaded it. Recorded the total magnification. So we're going to replace the amoeba whole mount with the penicillium. So I have that right there. And I'm going to start on the lowest setting, making sure I have everything in going under the light. Alrighty. Okay. down there. Be here all day. Okay. So this one's interesting. It has like a greenish, bluish, like strand. Oh yeah. With little, almost like microbe hairs coming off. It looks like a piece of yarn, almost. Like a very thin piece of yarn. Now I'm going to check it on the medium power. Oh yeah. Looks like a beautiful watercolor. Now we're going to go to the high power. Let me adjust this a little bit to see if I see anything. Again, with the high powered lens, I can clearly see there's something on the mount, but I can't differentiate what it is. The medium powered lens definitely has the best viewing of this. Now I'm going to remove, we're going to do the oil immersion. Okay, let me just make sure I'm not missing anything here. Repeat steps for them. Yep, so basically just doing everything, repeating everything on this. Okay, here we go. Let me add the oil. Okay. Turning this. Alrighty. Now, I don't know if I am just not doing this right or what is going on, but I'm having a really time seeing anything different when I use the immersion oil lens. Nothing really changes. Everything kind of looks the same. I'm doing a little bit of fine tuning here just to see if anything else pulls up. And I clearly see that there is little particles on there, but it doesn't really, I can't really differentiate what I'm seeing here. As far as shape, I mean, the shape is pretty pinpoint. So that's for that one. I'm going to remove this slide. Clean it with my lens. And we're going to move on to we're moving on to the yeast hole mount. Okay. Alrighty. Oh, I'm going to start with the lowest setting. Sorry, forgot. Okay, going back down. Okay. So on this one, it is showing up little green, tiny little bit of green. Very, very, very tiny little green bubbles clustered together. So I'm going to go to the medium power, and I'm going to focus this a little bit differently. So same thing with this one. But now I'm also seeing some red in there as well. Same formations, like little green bubbles clustered together. And there's a couple that are just independent. Go to high power. High power. Oh, high power. They look bigger. Can't see the little red ones anymore. You just see the, and this one doesn't look as it doesn't look as detailed. It's just like a basic green, little splotches. So I would say again the medium is the better viewing of all of them. So I'm going to go ahead and do the oil immersion on this one. Hopefully I'll get something going. Okay. Alrighty. Alrighty. And again, I'm not seeing really anything under the oil immersion. And I don't know if this is purposefully that I'm not seeing anything. It's the same grayish background with the little black spots that I see. No real detail. No, nothing on that. So let me get this off. The lens. Now we're going to go to the bacteria Spirillum. I'm going to start on my lowest setting. Okay. Get my okay. I gotta restage this a little bit. Alrighty. Okay. So for this one, it just kind of looks like clear colored little clustered formations that are spaced out. Let me try the medium. Medium, oh yeah, medium, you can see it way better. They look like little coiled pieces of hair almost in clusters. With a dark coloring. Like little, like moss kind of. So let me do the oil immersion lens. Oh wait, actually, sorry. No, I have to do the high power. Okay. So high power is not, I'll do the light a little. Not the greatest, surprisingly. I don't know if that's how these slides are supposed to be showing up, but I just see gray with a little bit of whitish background. And I do see the little the microbes, those are clear in color under this. Okay. So let me do the oil immersion. View of this. Oh Lord. What am I doing here? I think I screwed it in too hard. It's not wanting to come out now. Well, I think I did that too hard, so I'm gonna just take the medium powered out for time's sake here and replace that with this. Adding a little immersion oil onto the slide. Okay. And with the immersion oil, I am not really moving the coarse knob too much. I'm just using the fine coarse adjustment, and I'm not really seeing much again. Okay. Grayish again. Let me I have two more here. Put the medium back in. I don't know what's going on with my small one, why it doesn't want to come out, but I'll have to worry about that later. Okay, now we're going on to the bacteria Bacillus form. Alrighty. Starting with the lowest setting, I'm going to restage this, recenter this, I should say. Okay. There we go. So immediately I see clear microbes, not really any shape. They're just different sizes. Do this. No color. Okay. I'm going to go on to the medium power. And I see my eyes. Okay. Medium power again is winning. I see, it looks like it was gram stained with a little bit of that purplish colored one. And it's like one strand. And then there are different particles. Okay, then we're gonna go to the high power. This again looks like mossy. And in this one, you can see like little hairs or mossy, if you will. Okay, now, so I like the view of the medium again. So let me see here. All right, oil immersion time. Okay. Okay. Oil. I'm going to adjust the light on this one a little bit to see if anything comes through. It seems like there's a little bit more, has a little bit more streaking in this one, not so much circular structures, but it's the same color, like a light grayish, whitish background, and those streaks as well as those circular structures are dark gray. Okay. So the last one we're gonna do is the coccus, bacteria coccus form. Let me take this off. I'm afraid that oil is gonna get on my lens. Alright. Alrighty. Placing the smallest, starting there. Alrighty. Okay. Alrighty. So clear. Looks like watery. No real, I don't see any structures within those little clear water-like molecules. Let me go to medium power. There's like a lot of little bead looking things, like beads of water. That's for medium power. The medium is again the one I'm getting the best view on. Oh, actually, the high powered one. This looks like when I adjust it a little bit more with the coarse, it goes from dark to light. It looks like large groups of microbes, kind of elongated. Let me go ahead and do the oil immersion on this. Okay. And surprisingly for this one, the structure does look different in the oil immersion. The coloring is still the same. I don't really see any changes in the color, but it does look like the shape of the microbes are more noticeably different than in the other slides. But I still don't see any color. So let me make sure I don't have anything else to go over. You know, it looks like a kind of like a dirty window if I had it a good way. So it says clean up, store all the slides in the box, and I'm done. So thank you. Sorry about the long video.